Physiology of Nerve, Department of Physiology. Objectives. At the end of this chapter, the student will be able to define functional structure of neurons, list the two divisions of the nervous tissue, 3. Enumerate functions of neuron and neuroglia. 4. Explain membrane potential, graded potential, and action potential. 5. Discuss synapses and synaptic transmission. Outlines. Overview of the nervous system, functional structure of neurons, classification of neurons, and neuralgia properties. The membrane potential, RMP ion channels, action potential, nerve or nerve impulse properties, graded potential and action potential differences, propagation or conduction of nerve impulse, synapses and synaptic transmission. Overview of the nervous system. Among 11 body systems, nerve system and endocrine system play the most important roles in maintaining homeostasis. Nervous system respond rapidly to adjust body processes using nerve impulses. Endocrine system operates slowly and exerts its influence on homeostasis by releasing hormones that the blood delivers to cells throughout the body. Nervous system is an intricate, highly organized network of billions of neurons and even more neuroglia. Human brain has an estimated number of 10 power of 11 neurons and 10 power of 14 synapses. Principal cell types that make up the nervous system are neurons and neuroglial cells. Neurons, functional signal conducting cells specialized for sensory function, generation of thought, storage of memory, integrates idea, coordinates muscular activities. Neuroglia, supporting cells, 5 times outnumber neurons. The gay in the previous page, but an inordinate amount of them can multiply after maturation. Potential causes of glioma, brain tumor. Neurons, functional and structural units of the nerve system, specialized to conduct information from one part of the body to another. A neuron has 3 distinct parts. These are cell body or soma, dendrites, and axon. The basic nerve cells. This diagram shows the 3 distinct parts of neurons: dendrites, axon, terminal endings, synapses. There are different types of neurons, but most have certain structural functional characteristics in common. Cell body soma, one or more specialized slender processes, axon or dendrites, input region dendrites, conducting component axon, secretory or output region axon terminal. Soma, biosynthetic center of the neuron, contained nucleus plus most organelles, rough endoplasmic reticulum or RER per and knee cell body, responsible for synthesis of neurotransmitters or NTs. Neurofibrils maintain shape, structure, and integrity of cell. Multiple mitochondria. Why? Receptive service for interaction with other neurons. Most somata are found in bony embryos of central nervous system. Why? Clusters of somata in central nervous systems equals to nuclei and peripheral nervous system equals to ganglia. Neuronal processes, arm-like extensions emanating from every neuron. CNS or central nervous system consists of both somata and processes, whereas bulk of peripheral nervous systems consists of processes. Tracts equals to bundles of processes in the nerve. Nerve in the nerve or CNS. CNS equals to bundles of processes in the two types of processes that differ in structure and function: dendrites and axon. Dendrites, thin branched processes whose main function is to receive incoming signals, effectively increase the surface area of a neuron to increase its ability to communicate with other neurons. Their small mushroom-shaped dendritic spines further increase SA. Convey information towards the soma through the use of graded potentials. Axons, myelinated or unmyelinated. Most neurons have single axon or long up to 1 meter process designed to convey information away from the cell body. It originates from a special region of the cell body. That means axon hillock transmit APS from soma towards the end of the axon, where they cause neurotransmitter release, often branch sparsely, forming collaterals. Each collateral may split into telodendria, which end in a synaptic knob, which contains synaptic vesicles, membranous bags of neurotransmitters. The diagram looks like this. Axonema. Axon plasma membrane surrounded by a myelin sheath, a wrapping of lipid which protects the axon and electrically isolates it, increases the rate of AP transmission. This wrapping is never complete. Interspersed along the axon are gaps where there is no myelin. These are nodes of Ranvier. The myelin sheath is made by in the central nervous system and by in the peripheral nervous system. Functional classification of neurons. 1. Sensory or afferent neurons. It conducts impulses from periphery to the center. 2. Motor or efferent neurons. They conduct impulses from central nervous system to the peripheral nervous system. 3. Interneuron or association neurons. Integrative conduct impulses from sensory to motor area. Structural classification of neurons. These are 1. Multipolar neurons found in central nervous system, motor in function. Bipolar neurons, the second one, found in retina and inner ear, sensory in function. Unipolar neurons, the third one, located in ganglia of spinal and cranial nerves, sensory in function. The neuroglia, non-neural cells found in association with neurons, provide supporting functions to the nervous system. They are 1. Microglial cells, 2. Astrocytes, 3. Oligodendrocytes, 4. Ependymal cells, 5. Schwann cells, 6. Satellite cells. 1. Microglial cells. They are specialized immune cells that act as the macrophages of the central nervous system. Why is it important for the central nervous system to have its own army of immune cells? 2. Astrocytes. Star-shaped, abundant, and versatile. They provide nourishment to the central nervous system and are involved in the formation of the blood-brain barrier. 3. Oligodendrocytes. They produce myelin sheaths, which provide electrical insulation for certain neurons in the central nervous system. 4. Ependymal cells. They line ventricles of the brain and the central canal of the spinal cord. Some are ciliated, which facilitates the movement of cerebrospinal fluid or CSF. 5. Schwann cells from myelin sheaths around larger nerve fibers in peripheral nervous system, vital to neuronal regeneration. 6. Satellite cells, small cells that line the interior surface of peripheral nervous system. They regulate the external chemical environment. This is the diagram and their details. Resting membrane potential. All cells have a voltage difference across their plasma membrane equals to membrane potential or VM. Membrane potential, at least resting membrane potential, RMP. At rest, they are electropositivity out and electronegativity inside the cell, the neuron. What are the causes of the RMP? 1. Outward diffusion of K through K. Leak channels. ECF is very high in sodium, while ICF is very high in potassium or K. As a result, K is constantly leaking out of the cell. 2. Sodium plus or per K plus pump is constantly pumping 3 sodium ions out and 2 potassium ions in for every ATP used. Thus, more positive charge is leaving than entering. 3. Protein anions. Negatively charged proteins within the ICF that cannot travel through the PM. The diagrams looks like this. Selective permeability of membranes. Some ions permitted to cross more easily than others. Neuronal membranes contain ion channels, protein tubes that span the membrane. 1. Non-gated. Stay open all the time. 2. Gated. Open on the occasion of action potential, causing a change in the permeability of the membrane. Basic electrophysiological terms. Excitability. Ability of the cell to generate the action potential. Excitable cells. Cells that generate action potential during excitation. Inexcitable cells or muscles, nerve, secretory cells. Action potential is the marker of excitation. Stimulus. A sudden change of the internal or external environmental condition of the cell. Threshold or intensity. Lowest or minimal intensity of stimulus to elicit an action potential. Polarization. A state in which membrane is polarized at rest, negative inside and positive outside. Depolarization. The membrane potential becomes less negative than the resting potential. That means negative 70 MV. Repolarization. Membrane returns to the resting potential after depolarization. Hyperpolarization. Membrane potential become more negative than resting potential. In the bracket, negative 70 MeV. Electrical potentials. 1. Graded potential or GP. Local changes in membrane potential serves as short distance signals. Initiates action potentials if threshold is reached. 2. Action potential or AP sends signals over longer distances. Graded potentials produced by a specific triggering mechanisms or stimulus that causes the opening of gated ion channels, usually sodium plus channels, which open in response to stimuli, causing sodium ion to move into the cell or depolarization. Their initial amplitude may be of almost any size. It simply depends on how much sodium ion originally entered the cell. If the initial amplitude of the graded potential is sufficient, it will spread all the way to the axon hillock, where the gated channels reside. If the arriving potential change is supra-threshold, action potential will be initiated in the axon hillock and it will travel down the axon to the synaptic knob, where it will cause NT or neurotransmitter exocytosis. If the potential change is sub-threshold, no action potential will ensue and nothing will happen. Graded potentials vary vary in magnitude or strength. Stronger stimulus, more channels open, more positive charges enter, more depolarization. Longer duration of stimulus, longer duration of graded potential can be depolarizing or hyperpolarization. The diagram looks like this. Graded potentials lose strength with increasing distance from the site of origin. This is a diagram. Second diagram looks like this. Action potentials. Rapid, reversible, and conductive change of the membrane potential. Single action potential involves only a small portion of the total excitable membrane. No loss of strength as they travel along the membrane, or non-decremental propagation. Nerve signals are transmitted by action potentials. This is a diagram. If VM reaches threshold, sodium ion channels open and sodium ion influx ensues depolarizing the cell and causing the VM to increase. This is the rising phase of an AP, or action potential. Eventually, sodium ion channel will have inactivated. And K channel will be open. Now K ion, potassium ion of influxes and repolarization. This is the failing phase. Potassium channels are slow to open and slow to close. This causes VM to take a brief dip below resting VM. This dip is the undershoot and is an example of hyperpolarization. This is the diagram. The second diagram looks like this. Once action potential is initiated at axial hillock, no other triggering is necessary. Action potential triggers identical new action potential in adjacent area of the body. This is the second diagrams of action potential activity. Contagious conduction. AP is spread along every patch of the membrane down the axon. It is the third diagram. The worst yellow color is resting potential. The blue color is depolarization. The red color is repolarization. The conduction of axon potentials looks like this. All or none principle. Threshold or supra-threshold stimulus applied to a single nerve fiber always initiate action potential. Threshold depolarization equals to action potential. Supra-threshold depolarization equals to action potential. This is a diagram. Refractory periods. Period of decreased excitability following action potential. 1. Absolute refractory period. During the time interval between opening of sodium channel activation gate and opening of inactivation gate. Sodium channel cannot be stimulated. Sodium channel cannot be involved in another action potential until inactivation gate has been reset. 2. Relative refractory period follows absolute refractory period. Additional action potential possible with stronger stimulus. Stronger stimulus is more likely to cause additional action potential. Some sodium channels still inactivated. Imagine if you will a toilet. When you pull the handle, water pours the bowl. This even takes a couple of seconds and you cannot stop it in the middle. Once the bowl empties, the flush is complete. Now the upper tank is empty. If you try pulling the handle at this point, nothing happens. This is absolute refractory. Wait for upper tank to begin refilling. You can now flush again, but the intensity of the flushes increases. The upper tank refills. This is relative refractory. Myelination. Fibers covered with myelin sheaths, mostly lipid, at regular intervals along an axon. Once initiated, the speed that action potential moves down an axon depends on axon myelination and diameter. Myelinated, this is the diagram. Unmyelinated, slow, the larger, the faster. Myelin-forming cells, oligodendrocytes, found in central nervous system. One oligodendrocyte, several myelin sheaths on different axons. Schwann cells, peripheral nervous system. One Schwann cells, one myelin sheath on the axon. Conduction of action potential. If action potential is generated at axon hillock, it will travel all the way down to synaptic nerve. The manner of travel depends on whether neuron is myelinated or unmyelinated. Unmyelinated neurons undergo sweeping or continuous conduction of action potential. Myelinated neurons undergo jumping or saltatory conduction of action potential. It occurs in unmyelinated. Continuous sweeping conduction occurs in unmyelinated axons. The whole length of the membrane is depolarized. Velocity of conduction is slow. Consumes large amount of energy, analogous to diminish failing. Saltatory or jumping conductions. It occurs in myelinated axons, north of Ranver, only region where current FBW across the membrane exists. Region of concentrated voltage gated sodium channel. Velocity is faster. Economizes ATP. Rates of action potential conduction. 1. Which do you think has a faster rate of action potential conduction? Myelinated or unmyelinated axons? Myelinated axon has a faster rate of action potential conduction. 2. Which do you think would conduct an action potential faster? An axon with a large diameter or an axon with a small diameter? The answer to question first is a myelinated axon. If you cannot see why, then answer this question. Could you move 100 feet faster if you walked heel to toe, or if you bounded in a way that there were 3 feet in between your feet with each step? Answer for question 2 is an axon with a large diameter. If you cannot see why, then answer this question. Could you move faster if you walked through a hallway that was 6 feet wide, or if you walked through a hallway that was 1 foot wide? Synapse. Synapse is region where there is transfer of message from neuron to effector cell or post-synaptic neuron and the muscle. The junction between the first must be a neuron. Two types, electrical or direct. Second one is chemical or indirect. Electrical synapses, communication via gap junctions between smooth and cardiac muscle and some neurons of central nervous system. Provide fast, synchronized and two-way transmission of information. 2. Chemical synapses, communication via chemical neurotransmitters that diffuse across synaptic cleft. Provides slow, one-way information flow. The diagrams looks like this. Components of synaptic transmission. 1. Presynaptic terminal contains neurotransmitter or NT. 2. Synaptic cleft contains ECF and enzymes. 3. Postsynaptic neuron contains receptor for the action of neurotransmitter. The diagram is like communication between neurons at a synaptic junction. 1. Action potential arrives at a synaptic end bulb. 2. Depolarization of membrane causes the opening of calcium ions channels. 3. Increase in calcium ions inside of presynaptic neuron triggers exocytosis of neurotransmitter. 4. Neurotransmitter diffuses across synaptic cleft and binds to receptor or ligand gated channel on postsynaptic neuron. 5. Neurotransmitter receptor interaction results in either EPSCP or IPSCP. When neurotransmitter R combination triggers opening of ligand-gated sodium channels. This leads to membrane depolarization. EPSCP example: on nicotinic receptor. When neurotransmitter R combination triggers opening of ligand-gated potassium or chlorine channels, this leads to membrane hyperpolarization. That means IPSCP. Example: GABA on GABA receptor. 6. If depolarization reaches a threshold. 7. Action potential is generated on the postsynaptic neuron. Excitatory, vice versa, inhibitory synapses. 1. Excitatory, more likely to have action potential depolarization. 2. Inhibitory, less likely to have action potential hyperpolarization. Neurotransmitter. This chart shows in detail of neurotransmitters. Neurotransmitter removal. Why do we want to remove AKI from the neuromuscular junction? How was AKI removed from the NMG? Neurotransmitters are removed from the synaptic cleft via enzymatic degradation, diffusion, and reuptake. Properties of synaptic transmission. Unidirectional conduction. Synaptic delay 0.5 up to 1.0 microsecond. Fatigue, decrease in response of postsynaptic neurons after repetitive stimulation by the presynaptic neurons. Synaptic potentiation or facilitation, increase in postsynaptic responses caused by previous postsynaptic stimulation. Factors affecting synaptic transmission. 1. pH. Alkalosis, increase synaptic transmission. Acidosis, decrease synaptic transmission. 2. Hypoxia, decrease synaptic transmission. 3. Drugs, caffeine, theophylline, theobromine, increase synaptic transmission. Strychnine, increase synaptic transmission. Hypnotics, anesthetics, tranquilizers, synaptic hypnotics, anesthetics, tranquilizers, decrease synaptic transmission. Integration is summing of input from various synapses at the axon hillock of the postsynaptic neuron to determine whether the neuron will generate action potentials. The grand postsynaptic potential GPSP is the composite of all the EPSPs and IPSPs. A postsynaptic neurons can be brought to threshold by 2 ways. These are: 1. Temporal summation, rapid fringe of a single neuron. 2. Spatial summation, signals from different neurons. The diagram for integration looks like this: convergence and divergence. Communication between neurons is not typically a one-to-one event. Sometimes a single neuron branches and it is collateral synapse on multiple target neurons. This is known as divergence. A single postsynaptic neuron may have synapses with as many as 10,000 postsynaptic neurons. This is called convergence. Can you think of an advantage to having convergent and divergent circuits? Thank you. God bless.